This is Mrs. Popplewell again. We're going to continue looking at the nervous system with Chapter 11. The meninges are membranes that surround the brain and the spinal cord. It has three distinct layers. They are divided up into the dura mater and the arachnoid mater and the pia mater. The outermost layer is made up of white, dense connective tissue. It really has it splits into two layers. The first layer is almost continuous with the periosteum that is found on the skull. And so if part of the skull has to be removed, great care needs to be taken to tease away that dura mater because it's attached to the periosteum of that skull. Now it splits into the two layers where it encloses the dural sinuses, and that area collects venous blood from the brain in the dural sinuses. Now the dura mater forms partitions. It dips down into the longitudinal fissure here. It's called the falx cerebri, and it separates the left and right cerebral hemispheres. The falx cerebella separates the right and left cerebellar hemispheres of the cerebellum, where the vermis is found, and dips into the contours of these. The arachnoid mater is named for its web-like appearance in the subarachnoid space. It is the violet middle layer. It's the thin net-like membrane. Beneath it, beneath the arachnoid mater, lies a wide space called the subarachnoid space. This space is filled with cerebral spinal fluid. So you'll have cerebral spinal fluid found here, CSF. It serves as a cushion for the brain, and also with the cerebral spinal fluid to nourish that tissue. The deepest layer and also the thinnest, very delicate membrane in this salmon colored here, dips into all the contours of the brain, is the pia mater. It clings to the brain's surface. It is very thin and delicate connective tissue, but it has many nerves and blood vessels. It helps to nourish the tissue, outer layers of the brain tissue, and dips into the grooves. Now, I want to tell you two conditions that can happen that you need to be responsible for. Hematoma is blood that collects between the dura mater and the arachnoid mater. So if you hear that basically a brain bleed or hematoma, blood collects between the dura and the arachnoid mater. Now, you've heard of meningitis. Before you could enroll in school and be a student at this college, you had to get a meningitis vaccination. Meningitis is inflammation of the arachnoid and pia mater. Now, meningitis is an interesting term because it can be inflammation from either, but it can be caused by a bacterial infection or a viral infection. Bacterial infections are more easily treated because bacterial cells are different than our cells, but viral meningitis is much more difficult to treat because viruses are very close to our cells in nature. So any drugs that are used could be potentially harmful to our cells. So viral meningitis is more serious than bacterial meningitis. Both are serious, But again, meningitis is inflammation of the arachnoid and the pia mater. Here are the meninges of the spinal cord. Again, the outermost, the dura mater, and there's the arachnoid mater with the subarachnoid space. And directly on the surface of the spinal cord is the pia mater. Now, what's a little bit different about the dura mater here, remember the dura mater in the skull on this, in the area of the brain, in the skull, is continuous with the periosteum of the bone of the skull. But in the vertebrae, which is also bone, it is not continuous with the periosteum. It is not attached to the periosteum in the vertebral foramen, or the hole in the vertebrae. You have the same meninges as the brain, but again the dura mater does not attach to the vertebrae as it does in the skull. So you see all the different layers here. The dura mater, the arachnoid mater, and directly on the surface of the spinal cord would be the pia mater. Ventricles are interconnected cavities within the cerebrum. Okay, they're sort of C-shaped, these lateral ventricles. Okay, we just call these the lateral ventricles. They're ventricles 1 and 2, but we just named them the lateral ventricles. It is continuous with the central canal of the spinal cord. So as you go down here, here's the central canal of the spinal cord. And there are the lateral ventricles, and the third ventricle and the fourth ventricle. They are filled with cerebral spinal fluid. The lateral ventricles, third ventricles, fourth ventricles, and the cerebral aqueduct. And in addition to filling the subarachnoid space, the cerebral spinal fluid fills the ventricles, those interconnected cavities within the cerebral hemispheres of the brain and of the brainstem. The ventricles are continuous with the central canal of the spinal cord and have CSF. They're lined with epidermal cells. That's one of those neuroglial cells that we talked about of the central nervous system. Those epidermal cells are ciliated to help circulate that cerebral spinal fluid. So this almost looks like your hand as it forms like a C. It surrounds the thalamus and the hypothalamus in the area of the diencephalon. But this C shape here are the laterals, so there's 2 on either side. And this area right around the thalamus is the third ventricle. If you follow the cerebral aqueduct, it will lead down to the fourth ventricle, and that fourth ventricle almost protrudes posteriorly toward the cerebellum. Now on our model it would almost point toward the cerebellum, so that area is the fourth ventricle, and that cerebral aqueduct continues down and will become a little bit smaller and form the central canal of the spinal cord. Cerebral spinal fluid is secreted by the choroid plexus, and the choroid plexus are superior to the thalamus here. Most of the cerebral spinal fluid is secreted by specialized capillaries in the choroid plexus and in the lateral ventricles, ventricles 1 and 2. The cerebral spinal fluid circulates down into the third and then to the fourth ventricle, and then into either the central canal in the spinal cord or the subarachnoid space of the meninges. Cerebral spinal fluid is reabsorbed back into the bloodstream through the arachnoid granulations. And these subarachnoid granulations project into the dural sinuses. Cerebral spinal fluid movement is aided by the cilia of the epidermal cells. The total volume of cerebral spinal fluid is about 140 milliliters. Okay, what is a milliliter? Well, there are 1,000 milliliters in a 1 liter drink. The average water bottle is about 500 milliliters. So actually 500 milliliters, about a half a liter, is secreted daily to replenish the circulating 140 milliliters, and it's replenished or replaced about every 3 to 4 hours. So about 500 milliliters is produced in a day, and then about 140 milliliters is circulated at any one time. It functions as a mechanical protection, so it cushions. It has chemical protection. It helps to regulate ions, hormones, etc. It helps to form the blood-brain barrier. The brain is sensitive to extracellular changes, so the cerebral spinal fluid is more tightly regulated than the composition of other tissue fluids. The capillaries are relatively impermeable. Nutrients come in by facilitated diffusion or active transport, and it's very, very selective. Most toxins or drugs are not allowed, and those things that are allowed to pass must be lipid soluble. Okay, again, cerebral spinal fluid is secreted by the choroid plexus, and the choroid plexus are found superior to the thalamus here and into the third ventricle. Circulates in the ventricles and the central canal of the spinal cord, and then the subarachnoid space. It completely surrounds the brain and spinal cord. It is a clear liquid. Now, when they do a spinal tap and take a sample of cerebral spinal fluid, that is one thing they look at immediately. As soon as they draw that CSF into that syringe, they look at it, and it must be clear. It will look like water. If it is pink, then there is a hemorrhage, blood in that cerebral spinal fluid, some type of injury. If it is cloudy, then there's a growth of microorganisms, bacterial infection probably. Okay, so that's pretty serious. Nutritive and protective. It helps maintain a stable ion concentration in the central nervous system. The spinal cord structure. It extends through the foramen magnum. As soon as, right here is the brainstem, and as soon as the brainstem comes out of the foramen magnum, it becomes spinal cord. It extends from there to the second lumbar vertebrae. Here's the brainstem, and this would be the area of the foramen magnum, and there's the cervical enlargement and the lumbar enlargement. It's about 17 inches long. It contains 31 segments and therefore gives rise to 31 pairs of spinal nerves. There are cervical and lumbar enlargements. Now, this area here is the cauda equina, or horse's tail, and you go further and more inferior you get, the phylum terminale represents the distal portion of that tail, and it actually becomes pia mater. It's a very thin, delicate, just membrane there. Now, before we talk about the spinal cord, let's look at the structure. You went over the model in lab. Let's just review the different structures. We're going to use our directional terms here and add some other terms to it, and it should make it a lot easier to identify the structures. This drawing looks very similar to the prepared slide you looked at of the spinal cord, cross-section of the spinal cord. You have white matter where you have myelinated nerve fibers, and then the gray matter where you have unmyelinated nerve fibers. Posteriorly and anteriorly you have various structures. So you're going to use those directional terms, the lateral, anterior, posterior. Now we use those terms for the structure of the spinal cord itself. If we're talking about the spinal nerve and the roots of the spinal nerves, for the posterior side we use dorsal instead of posterior, so be careful on that. So this is the dorsal root of the spinal nerve, and anteriorly we have the ventral root of the spinal nerve. And in this area we know this is the dorsal root because we recognize that ganglion. That ganglion is a bundle of soma or cell bodies of that dorsal root of the spinal nerve. Okay, so let's use the directional terms and name the parts. Now, white matter is called funiculus. Funiculi is plural. Alright, so if we're naming a particular part, then we would use funiculus. This area is posterior, so this would be posterior funiculus. Okay, lateral funiculus. If we were talking about both sides, it would be lateral funiculi. Anterior funiculus. Okay, dividing the right and left halves are these grooves. The most prominent and deep groove, now let me tell you the difference between a fissure and a sulcus. The fissure will usually divide something that you could separate very easily because it's a deep groove. A sulcus is a very shallow groove. It's not as prominent, so you don't have much of an indention here, but you have a pretty good indention here where you have this fissure as opposed to this sulcus. Anteriorly we have the anterior median toward the middle, median fissure. Posteriorly we have the posterior median sulcus. Okay, so now let's name the gray matter, that bundle of unmyelinated nerve fibers. We're going to use our directional terms again, and it looks like an upside down butterfly, the wings of a butterfly here. This would be the posterior horn. This would be the anterior horn, and if you had a little bit more of a prominence there, that would be the lateral horn. Now in the center of this gray matter is this hole, and this is the central canal of the spinal cord. Okay, the central canal, that would be lined with epidermal cells that are ciliated, that helps circulate that cerebral spinal fluid. Okay, the right and left halves of the gray matter are joined together by the gray commissary. Alright, so they are ascending and descending nerve tracts. Remember, in the central nervous system, a nerve is called a tract. So we have ascending going up to the brain and descending nerve tracts. But you also have innervation going from one side of the spinal cord to the other across the gray commissary. So you have ascending and descending tracts, but you have these that go back and forth here, sort of a communication between the right and the left halves of the spinal cord. The tissue here is predominantly interneurons. Remember, we talked about interneurons makes up about 99% of the neurons in the central nervous system. The posterior half receives information from affectors, the dorsal root. Okay, if we extended this on down to all the receptors down here, it could be from organs, so they'd be visceral afferent receptors, or it could be from muscles, glands, those sensory receptors found in the epithelial tissue, hot and cold receptors, pain receptors, light touch, deep pressure. Okay, so that sensory information comes in from those receptors through the dorsal, through the spinal nerve. Now here it would be spinal nerve, and then it splits off into two parts, the roots. So it'd come through the dorsal root ganglion, through the dorsal root of the spinal nerve to the posterior funiculus, to the posterior horn, and then it's more or less sort of kind of interpreted by these interneurons. If it were a reflex, the reflex reaction does not go to the brain, so that interneuron would not channel that information to the brain. It would actually go to the anterior horn, to the anterior funiculus, to the anterior or ventral root of the spinal nerve, to the effector organs, if it's a reflex, to the muscle, and the muscle would be the effector. Okay, efferent would be that path, effector would be the organ that responds. Okay, this would be afferent or afferent, and afferent affectors, okay, would be those sensory receptors that send in the information. Okay. Again, white matter is myelinated nerve fibers, and then you have tracts, and you have that send the information ascending, descending, and then from side to side. Spinal cord functions. It's the center for spinal reflexes. It's the conduit for nerve impulses to and from the brain. Now I'm just going to go over this pretty quickly because you looked at this in lab. Reflex arcs. Remember, I told you that the reflex doesn't have to go to the brain to be interpreted. You have the doctor uses a reflex hammer and hits that knee, and those receptors, actually there are receptors found within the muscle, not just the skin, but within the muscle, that responds to that stimulation. That information is carried on the sensory neurons. This is the affector. The receptors, if it's a reflex, it would be in muscles too. So the muscle would be the affector. The receptors are called the affectors. This would be afferent or afferent, going through. Now remember that all spinal nerves are mixed, taking information to the spinal cord and from. Okay, so then it splits to the dorsal root that carries the information. It passes over that cell body of the sensory neuron does, and then through the dorsal root, through the dorsal funiculi, just the short way, to the I'm sorry, not the dorsal funiculi, but the posterior funiculi, the posterior horn. The interneuron interprets the information. Since it's a reflex, it doesn't go to the brain, so it takes care of it right there in the spinal cord. If with the response, response would be efferent to the effectors. Okay, so it'd go from the gray matter or the this interneuron through the anterior horn, through the anterior funiculus, through the ventral root to the effector muscle or gland for the response. Now, if it's not a reflex, then it is taken to the brain to be interpreted. Reflexes are automatic subconscious responses to stimuli. I'm just going to talk about this for just a second. A cross-extensor reflex, the flexor muscles contract. The flexor muscles on the opposite sides are inhibited. The extensor muscles on the opposite sides contract for balance. So what this is saying is that reflexes have a purpose. The purpose of these reflexes is to help with smooth muscle movements. So can be in walking or stepping, all for smooth coordinated muscle movement. And if there is a problem with these reflexes, you would have a problem with smooth muscle movement for smooth gait or walking the stairs. Okay, so that's just an example of how these reflexes work. Okay, tracts of the spinal cord. You have ascending tracts that conduct sensory impulses to the brain so they can be interpreted, and then the information response to that comes from the brain on the descending tracts. And again, tracts are just nerves within, bundles of nerve fibers within the central nervous system. The descending tracts conduct motor impulses from the brain to the motor neurons reaching the muscles and glands. Okay, and these are the areas of the different tracts. Now, you are not responsible for naming any of these tracts, but you are responsible for knowing what a tract is and that there are ascending, descending, and then remember the gray commissary have tracts that go this way to help to communicate across the spinal cord. You have it in the white matter, myelinated axons that run in 3 directions, ascending and descending, and then that would be in the white matter, and then unmyelinated in the gray matter. And the commissural are the ones that run from side to side, from one side of the spinal cord to the other side, so you can have communication that way. So 3 ways of running that information, 3 directions. Okay, let's